All right. Oh, that's gonna like echo on me, huh? All right. Well, thanks everybody for joining here. Listen to me talk about ducks, conservation, and so forth. Title of my talk: Manhandling mallards, conservation paradox of feral populations and the meaning of wild. A bit, just so you know what I do. So I am sort of like Sarah. I'm a geneticist. I'm a wildlife geneticist. And just like Sarah, we sort of do everything under the sun. We do everything from habitat assessments to, capturing, banding, telemetry units on ducks, understanding movement ecology. But the underlying theme in my lab is genetics. And that is because whether I'm asking the question in evolutionary senses or whether I'm trying to describe what is occurring to a management agency, it's really asking the question of what are these things? If we don't actually know or we assume things that are wrong about our study organism, then our models, our assumptions, our biases are going to be invoked in what we are going to, the story we're going to tell about that organism. And so what I come in is I use genetics to identify, is this parent one? Is this a hybrid population? Is it parent two, parent three, whatever it might be? And by doing that, understanding population structure, identifying ancestry of those individuals and so forth, we can really then guide conservation, at least in my opinion, in a more meaningful and more direct way. Among those themes that I study is hybridization. And for a long time, life, we believed life just basically bifurcated through time, and essentially hybridization was just like this one-off situation. But as we continue to gather more and more genetic information on lots of non-model organisms, we're finding that hybridization is actually quite prevalent, not only in the past, but in today's time, including humans. And in fact, if you ask me, I would say the Homo sapien is a hybrid species itself, because we are the combination of Homo sapien, Neanderthal, and some of us have Denisovian in us, right? And so technically, we are an interbred lineage of multiple Homo sapien and other ancient man. Now, this is the part where I want to make sure everybody understands the difference between hybridization and gene flow. So hybridization is when parent one and parent two breed and make this hybrid. It's called an F1 hybrid. It's the first generation. If that hybrid is inviable, like a mule, or doesn't survive for whatever reason, that's in biological sense and in management sense, that's lost breeding potential. The hen that chose wrong created a clutch that is no longer viable for the population. That's lost breeding potential. You have hybridization. What gene flow is that you have hybridization, but that hybrid, that F1, then is able to breed back into parent one or parent two, and thus moving genes from the two parents. F1 is essentially this microcosm of genetic information from different lineages. All right, so now that everybody understands the difference between those two, let me introduce you to the system that I work on. Now, I will note that while my system are waterfowl, and particularly mallards, my group uses the same exact techniques and methods and analyses to answer questions from everything from snails to whales. They are, genetics, anything that's got DNA and a bit of funding is what we do. So the mallard complex, hopefully everybody here knows who the mallard is. King mallard up there. So they have a Holarctic distribution, naturally found across Eurasia and North America. But then there's actually 14 of these mallard-like ducks. This is called a successful rapid radiation. They evolved, diverged over the last million years. And there's a mallard-like duck found pretty much on every continent and island. So in fact, in Africa, you've got 3 of them. Australia, the South Pacific has a couple subspecies of that Pacific black duck and Philippine duck. And in North America here, on the Hawaiian Islands, we have this thing called Hawaiian duck. Well named, Laysan duck on Laysan Island. And then on mainland North America, in addition to the mallard, we have Mexican duck, mottled duck, and American black duck. Now, so that's my intro to the organism. One of the things that we are now finding, and particularly because mallards are aptly moved by us throughout the world, is that this introductions and secondary contact is increasingly causing anthropogenic hybridization. And what that is either direct or indirect effects by people moving things around or changing habitat that causes species that were once allopatric or separated to come into contact. That is anthropogenic hybridization. A big part of that is the introduction of captive bred or domestic variants of the wild thing. If you don't know this, I have to introduce you to another complex of mallards, and that is the domestic mallard complex. If you go to your favorite urban park and you see a bunch of fancy looking ducks, those are actually all just mallards of European descent. They were domesticated at 500 BC, sometime in somewhere in China and Asia around then. They were the last sort of successful big domestications, with wolves being the first, ducks being the last. And particularly out of all those guys there, including the Peking duck, is this thing called the game farm mallard. While all those other ones are being bred for consumption, agriculture, egg production, meat production, whatever it is that you're looking for, fanciness, just like the wolf dog thing, you could have a Chihuahua from a wolf, is this thing called the game farm mallard. The game farm mallard is distinguishable from the rest of them is because they are a breed that was directly created by King Charles II, as far as I understand, for the propagation for hunting, for augmentation of wild populations. And that game farm mallard that was created somewhere in England has been propagated and moved across the world. And they've been introduced everywhere. They've just been introduced everywhere. In Eurasia, this is where the origination of mallard derivative domestics occurred. About 500 BC, game farm mallard strains were created in the last 2 centuries via selective breedings. And today, somewhere in the range of 3 to 6 million game farm mallards are now being annually dumped by their respective fish and game associates to maintain a fall flight. In North America, somebody had the same idea as the Europeans, and the Atlantic Flyway decided, we're not happy with just black ducks, we would like to see greenheads too. If you don't know this, mallards were actually a one-off east of the Mississippi River for a very long time. And on the Atlantic Flyway, somebody had the idea that they should start dumping mallards out there. Initially, they did try to do captive breeding of our own mallards. That didn't take. It takes time to get wild birds to breed the way you want them to breed in a captive setting. So somebody else went on a boat to England and went and got some birds and came back and started to propagate them. State, federals, and private citizens were dumping birds of about 500,000 a year from 1920 to 1960, and from the 60s to today, about 250, 210 is coming out of within the Atlantic Flyway. I would say those numbers are lower than you think. In the South Pacific, game farm mallards were brought in, imported for food and sustenance by Europeans in the 1850s. Of course, they got out, and now there's a large feral population that is causing genetic extinction of the Hawaiian duck. Multiple feral populations. In New Zealand, this is a success story. They brought in 30,000 game farm mallards, from the 1860s until about 1950. They stopped there. 30,000 mallards have turned into 5 million, a feral population of 5 million. It's quite successful. All right, so that is the history of what I'm about to show you. The methods that I use to understand genetic diversity is called DDRAT. I'm not going to bore you about this. If you really care of how to do this, you can ask me later. But in essence, in a snapshot, I can look at thousands of markers for hundreds of individuals, and I get a nice representation across the genome, thousands of markers. I can do everything from population genetics to looking for loci under selection and so forth and so on. So if you're used to microsatellites that require you about 10 years to actually get them developed, hope for a few of them to be actually polymorphic, and eventually being cheap, this is a much faster route for anything that you are just starting out to figure out. And what did we learn about these? So I study all of these locations in North America. The introductions, and you're going to be, this is what I'm going to focus on later on, is North America. But in North America, the introduction of game farm mallards has resulted in widespread hybridization. Hawaii, you can see these are PCA plots of individuals. Each one is like different types. So you can see the things that are intermediate in space between major groups are hybrids. So just so that way you understand that. So you can see hybrids of Hawaiian ducks. So the introduction of these domestic mallards in there resulted in lots of hybridization. And same thing in New Zealand. These are all based off of hundreds of samples. So New Zealand, you can see the nice thing is we can find New Zealand gray ducks, but then you can see that there are mallards, this thing called New Zealand mallard that we coined, and lots of things in the middle, right? Lots of hybrids. All right. So we can show, my lab has been showcasing that the introduction of these things, not only are game farm mallards, while they might look like a mallard, they are not a mallard. And hopefully by the end of this talk, you will agree with me. So let's go to North America. So in North America, we, for a very long time, I was under the dogmatic principle that game farm mallards, it's okay to put them out because they don't really breed, they don't survive, they don't breed, it's perfectly fine. Until I started doing these genetic analyses and started to find this secondary genetic signature on my, in my mallards. And that signature was very defined by location. As I went from west to east in North America, I continued to find a secondary genetic signature. What you're looking here is at mixture plots or ancestry. If you ever did 23andMe, this is what this is. Essentially, if you're one color, you're part of that group. If you're multiple colors, you have ancestry of multiple lineages. And that's what this means. What I wanted to make sure is that that secondary grouping is not park duck. So what we did was we, while we were catching Mexican ducks, we also caught these things called khaki Campbells, another domestic variant. And they are my proxy for park duck, this thing. But you can see that park duck and game farm mallard are two different colors. So their genetic ancestry is completely divergent. And that genetic ancestry is completely divergent from that lighter gray color, which is our wild mallard signature. And so as you go from west to east to the Atlantic Flyway, you increase in game farm ancestry. Hopefully everybody's with me. Now, somebody came up to me and said, well, maybe our Atlantic Flyway mallards have always been like this. So I said, okay. So I went to the Smithsonian and I collected ducks, black ducks and mallards. This was a black duck mallard kind of question at the time. Lo and behold, it turned into a mallard question, of course. And so I got birds from 1860 to 1910, before any of these like large scale releases occurred. And I wanted to ask two questions. Well, were mallards and black ducks always the same? And two, are today's Atlantic Flyway mallards completely different than what was here 150 years ago? And what we found out is that historical mallard or historical black ducks and today's black duck, what is a good black, genetically a good black duck, is identical. So yes, we still have black ducks. The other part of this story, oh, did it freeze? There we go. So mallards, this is historical mallards and today's mallards. Now, if you will note that today's mallards and historical mallards are only the ones in western flyways, Mississippi Flyway and Central and Pacific Flyways. These are your today's Atlantic Flyway mallards. That means they are have been genetically transformed into something completely different because of hybridization. They are no longer the same thing as 150 years ago. So the history, there's a lot more there for the black duck, but you have to just assume I'm correct because this is published. Is that the American black duck diverge is not just this thing that diverged from a brown duck, they diverged from mallards. And so they had a allopatric history for a long time with a bit of bouts of gene flow here and there naturally, until 1920 when we started to dump those birds onto the landscape. And remember, people wanted to shoot greenheads, so we put out males, primarily males, and that caused a lot more gene flow. Now, what we have today in North America, we still have black ducks, and I believe as long as we have boreal forests, we will continue to have black ducks because it appears that mallards do not cannot efficiently mate and have clutches in good boreal habitat. We still have wild mallards, which is great. We also have this thing called a feral wild mallard hybrid swarm, which is essentially largely in the Atlantic flyway, but what I'm about to show you is worldwide. So from that story, I wanted to understand what are the genetic consequences from introducing domestic game farm mallards worldwide, right? So we know these histories, it's well documented. So I was like, what's going on? To do that, we went and we got about 2,000 samples. All throughout Eurasia, North America, New Zealand, and Hawaii are feral populations because we know there were no mallards until we put them there. We know the origin, well, now we know the origins of them. We have heavy sampling of the Atlantic and Mississippi flyway with Central and Pacific a bit less, but still pretty good because we kind of lump them together. And then known game farm mallards at this time, we had Kentucky, New Jersey, South Carolina. We just added Illinois and Georgia. I will note that they are of the same origin. And then also known game farm mallards from Ireland and Sweden. So we got all that good stuff. You can see my map of all the nice samples. We wanted to understand lineages based on a couple different marker sets. One was the mitochondrial. One was the mitochondrial DNA, and the other one was nuclear-based DNA. Everything you're looking at is almost 37,000 SNPs, so single nucleotide polymorphisms. So you can just, that. Some of the analyses that we did was we looked at evolutionary, you're going to see a bit of evolutionary history with tree mix. We looked at demographic histories, although I think I deleted that from this talk because it gets too muddy. But then we also did this thing called gradient forest. We wanted to understand whether mallards are adapting to urbanization. All right, this is mitochondrial haplotype network. Each ball is a different haplotype. The size of them is the total number of samples in them. Each color denotes a different group. And what you can see here is that they're split between these two known haplogroups, which is Old World and New World. Only one mallard in China was found to be possessed a New World B haplogroup, which was likely the origination of hybridization with yellow-billed ducks. No, not yellow-billed ducks. Indian spot-billed ducks. There it is. Chinese spot-billed ducks that also possess a B-like haplogroup, or a mallard actually went to China. Domestic and feral mallards harbor fewer haplotypes. There's only 6 major haplotypes that I can find in North America. Majority of Europe, all of New Zealand, Hawaii. Yeah, so what you're seeing here is that a very small, so what you're seeing in the B haplotype is wildness. Lots of unique haplogroups. So something on the right is the diversity you expect under something of a wild system. The thing that you're seeing on the left, the Old World A, is what happens when you have bottlenecks, and then you spread those bottlenecks around. You can see khaki Campbells here in black, their own haplogroup, and one of our Russian samples, I never got pictures of them, but actually it turned out to be a khaki Campbell that they thought was a wild mallard. Over 90% of all Old World mtDNA introgression that is found everywhere outside of Europe can be described in these 6-7 haplotypes of Old World A. And when we plot this on a map, this is what it looks like. You can see Europe, Eurasia is pretty much all A. And what you're seeing is that there is a general sense of decreasing A haplotypes from east to west in North America, which is exactly in line with where we dump game farm mallards. Now, moving on to the nuclear signature. Remember, we did 37,000 SNPs, single nucleotide polymorphisms, about 1,900 samples, and we plotted them all out here. The nice thing here, feral game farm mallards from Europe and Eastern North America, the things that we are dumping are the same thing. They overlap in genetic space. Wild mallards of North America and Eurasia, also the same thing, although what I'm going to show you is very small signatures of differentiation. Greenland mallards, something unique, completely different than the rest of the groups. They come out with ancestry mapping. They also look completely different. This was probably a founder event, and that Greenland has its own little pocket of mallards. All remaining domestic lineages are within this group, and they all diverge because of various forces of artificial selection and differentiation in those groups. Wild and domestic hybrids recovered were largest where the largest releases are. Now, before I put this on a map, I want to just show you a bit of this phylogeny, looking to understand where do all these groups come from. So you have these two main lineages, wild and domestic, which is nice. The most basal group is that wild origin, so the wild mallard is the ancestor to the domestic groups. Eurasian GFM ancestor, so the game farm mallard ancestor is the ancestor to those that are being released in North America, the feral population in Hawaii, the feral population in New Zealand, and the gene flow groups that you see, these little arrows, are actually consistent with what we know because we have two forms of introductions into New Zealand, a more recent one, and then that game farm mallard 2 is the South Carolina lineage. And that lineage, I was given information that somebody in the 1980s went out and caught wild birds specifically to breed into their population. And I was like, okay, that's fine. I don't believe you, but it's true. They did. That means they broke Migratory Bird Treaty Act. This is also a plot. This is an ancestry plot, and I'm gonna walk you through it. This is the last tedious slide, so to speak. The main thing is that game farm mallards is the same exact genetic stock being released in Eurasia and in North America. So you can see that they're all that red. Now, what you could also see is that Eurasian game farm mallards have wild in them. And that's because your game farm mallards in Europe purposefully leave their game farm mallards out so that way wild birds interbreed with them during the breeding season. You could also see that third group, this guy right here with a bit of North American wild. That's that South Carolina group that directly went ahead and caught wild birds and interbred into them. This here is the wild population. The one thing that we now can show with our SNP data set is that European and North American wild mallards do have statistically different frequencies in their genotypes. That's why Europe is that darker green and North America is that lighter green. That's the overall signal. Hybrids equal game farm mallard, wild mallard everywhere. There is, while we have khaki Campbells in here and other things, the thing that is found to be of hybrid origin is always wild with game farm mallard. Feral mallards, this was actually a shock. I thought they'd be park ducks, but actually they were also game farm mallards. So we know that they are, so game farm mallards are the origin to that location as well. As the same thing for New Zealand, although they've diverged ever since due to hybridization with New Zealand gray ducks. That's a different story though. So what I'm about to show is based on this, this ancestry mapping. I created a cutoff. Pure wild mallards are anything that are above 90% assigned to the wild cluster. Feral game farm, anything over 90% assigned to game farm cluster, and hybrids are everything in between. And that is what that looks like on a map, right? And so hopefully everybody can see where the hybrids are. And that is your epicenter of hybridization, right? So these locations, Central Europe, particularly France, who put out about 3 million birds a year, the Atlantic Flyway, who still do 200,000+ birds a year, are where we keep finding hybrids over and over. Now, what I also wanted to understand is, okay, so there's all this hybridization. How does this explain some of the adaptiveness, or is there adaptive differences? So we used a program called Gradient Force. It's a machine learning regression program that identifies associations between genetics and environmental factors. We looked at bioclimate data, various data variables, and you can see the descriptions, everything from temperature, precipitation, elevation, complexity of vegetation, and so forth. And on top of it, we also put these two indices at the bottom, human footprint. These are indices of urbanization. I'll just cut to this part. So what you're looking at is the genetic space of it's like niche. It's a niche space of these different forms and where they're found. So what's interesting here is that Western North America and Eastern Europe, where the least amount of hybridization is occurring, have the same niche space, that wild niche space. When we look at feral populations, they actually are found in a couple different locations. For Hawaii, we need more samples, but New Zealand's got that sort of location that also overlaps birds out of southwestern Eurasia. But this is the most interesting, is that Western Europe and Eastern North America share the same genetic niche space. These are locations with the highest amount of domestic integration and urbanization. So there's something that is interplaying in these birds that are causing them to start to shift in adaptive space. How that translates into the future is a paper we're working on right now, and hopefully you'll read it at some point. So populations showing local ecological adaptation that appear to be human linked. All right, so now I sort of gave you that big story. This is the next part where we dive into North America to understand what's occurring in North America. So we've been able to, with the help of state and federal agencies, collect over 1,000 samples in North America across all the states, obviously almost every state in the Atlantic and Mississippi Flyway, with a bit of all of the provinces of Canada. And then, yeah, we sort of petered out in the West because that wasn't our primary question at the time. But what you can see, and another thing is we have a fancy way of actually identifying early hybrids, so F1s, F2, F3, and then the things that they're hybrids, but we don't know what age they are. That's what all those color forms are. So what's occurring? So early hybrids, so these F1, the prevalence of F1s, F2s, F3s are exactly where you expect them to be, which is where the largest releases are, which is New Jersey, basically. That's the epicenter. Most hybrids are unidentifiable. That means they're generational hybrids of hybrid descent. That is essentially a hybrid swarm. Now, based on our estimates and our over 1,000 samples in the Atlantic Flyway, if you go somewhere in the Atlantic Flyway and look at or shoot a mallard, you have a 2% chance that it is a wild mallard currently. Keep that in mind. Hybrid prevalence decreases east to west, as you expect. So there's a petering out as you go east to west. Interesting patterns that may explain some, maybe due to behavioral differences of these types, are found here. And what's interesting is I made up these hypotheses strictly based on what you're about to hear, strictly based on genetics. Didn't know what these birds did, but we now have telemetry units out that actually show I was somehow right. So the first one is this. In this mixed pool of sea of hybrids, you have these pure strains, right? You've got Dakotas. You guys here have pure mallards. And funny thing is that the southern part of the Mississippi Alluvial Valley also has pure mallards. So I hypothesized at the time that that is the feeding source from that location to that. Then you've got the Great Lakes region that is declining in population size, just like the Atlantic Flyway, so the Great Lakes region, Atlantic Flyway, showing similar patterns. Hybrid prevalence is actually quite high. In fact, If you look at Michigan, we're finding early hybrids there. Somebody must be releasing birds, or birds from the Atlantic flyway are coming into those locations. So with those hypotheses, I worked with several folks you're about to see on specific studies of locations. So this is the lower Mississippi alluvial valley. Those are the states. And what you can see is based on my smaller data set and based on this much larger data set, generally with the nuclear genetics, just look over here, generally they're pure. About 4% of samples are of hybrid origin, and all of them are like, you know, only 15% hybrid. You know, 15% of the genome is explained by game farm mallard genetics, which is pretty good, today's standards. I then said, hey, let's see, I partnered up with Doug Osborne out of the University of Arkansas at Monticello. He's putting telemetry units on hundreds of mallards every year. I said, hey, Doug, send me some blood. He said, yes, Phil, let's do that. And for Arkansas, you know, whether I did it with 250 samples or 200, still very low prevalence of hybrids, and all of them very late back crosses. So we're like, where do those birds go? Oh, they go to the Dakotas. That's amazing. So the same locations, the majority of those birds are going to the prairie pothole region. That is the source of the lower Mississippi alluvial valley. All right, so that hypothesis is quite validated. That's interesting. So then what about the Great Lakes birds? For the Great Lakes birds, I partnered up with Michigan State University, a PhD student there, Ben Lukanen. This is the telemetry work is part of his stuff. And of course, I did my genetics. And so let's plot that out. Way worse, right? The Great Lakes is a mess. So about the complete flip, 60% of those birds are hybrids. And a lot of them are actually early stage. The only place I ever find this many hybrids is like New Jersey and France. And so something obviously is happening there. So then we're like, what do these birds do? So let's put some telemetry units, and they do this. They don't want to go past Ohio. They just make these like weird migratory behavioral most of them stay within urban settings. They don't want to leave. They sort of move about, but they will not cross that line, right? And so there's behavioral differences of these very distinct, genetically distinct stocks. And so hopefully you can appreciate some of the mechanistic behavioral differences with the telemetry stuff, but there are many more things. The first thing is the fact that game farm mallards and wild mallards are 5 to 10% different genetically. In the bird world, that would be species level. Let me just tell you that an American black duck and a mallard is 1.5% different. So this is wildly different. Growth rates are completely disproportionate in these birds. Game farm mallards are bred to be lighter. They're 900 gram bird versus a 9 to 1200 gram bird of a wild one. Again, you have a smaller bird. Fertility is quite interesting. These birds, if you put them on Purina chow, so we have a study that I'm going to show you some data on at Forbes Biological Station where we have birds, we're doing these feeding trials with them. And, you know, these game farm mallards, if you just keep feeding them Purina chow, they keep pooping out eggs. They do not actually make nests. They just walk around and put eggs on the ground. Again, good stuff in a domestic setting, not so great in the wild, I imagine. The wild one, the other part of this is that they also, so our European counterparts have shown that they have about a 5% survival rate as compared to wild mallards out of France and Sweden. That study, the band recovery study done there. They don't, and hopefully some of that telemetry stuff shows that they're not doing regular wild migratory behavior. The other part of this is their morphology. Their morphology has been changed, and what I'm about to show you, to pecking, essentially. Game farm mallards, what we show have these smaller, stubbier looking bills with their lamella densities, so their little teeth that allow for filter feeding have spread out. And you can imagine in a cage where everybody gets to eat bread, corn, large seeds, there's no more selection for filter feeding, but in fact, artificial selection for pecking. And that's what appears to be happening to game farm, what has happened in game farm mallards. Again, our European counterparts also published on this, and we're about to show this in our own. And what you're about to see is that translates to wildly different feeding efficiencies. So this is part of this project with SUNY ESF, Mississippi State, Forest Biological Station, myself, and funded through the NSF. We took a whole bunch of birds. I genetically vetted everybody. We had pure wild, known game farm, a few hybrids, and we did morphological analyses as well as feeding efficiency. This is the first set of morphological data, preliminary, but we just finished all these analyses. I just don't have the most complete charts. But what you can see is that game farm mallards are no longer proportional in size. So what happened is that they have smaller wings and longer legs. And in evolutionary histories, when we see that, that is a bird that is going towards more walking, right? This is what we see in Hawaii a lot of times, or in New Zealand, where a bird comes and it becomes less and less flighted. Those smaller wings, we are looking into flight efficiency, and what appears to be that they are actually better at fast flight, quick flight, but non-sustained flight. So they cannot actually move large distances, potentially explaining why we see those behavioral differences from those Great Lakes birds. For the bill, what we see is this, that they basically have the culmen length, culmen width, and bill height are all statistically different. So game farm mallards have shorter, wider, and taller bills than wild mallards. And this is actually more consistent with the goose-like bill. And so we want to understand morphologically, so we have this morphology differences. How does that translate mechanistically to actual feeding efficiency? And I'll just, what we did were these feeding trials, put a GoPro, we had these birds on specific diets. We then sifted through those diets, looked at dry weight of what was left over compared to what we put on, and then, yeah, had a good time. And this is what we found, that game farm mallards had 50%, whether it's male or female of the other sex, had 50% decrease in feeding efficiency. That means that they needed twice as much food and twice as much time to gain the same calories. Now, here's the kicker. The thing that creates your population is the worst, right? Female game farm mallards had the lowest efficiency in feeding. So you can imagine now, if you put this bird into nature, that these traits are not so great. And this generally explains this, is that once you start stocking populations with groups of individuals where everybody gets to win, they will start to put these recessive traits, what we call maladaptive traits, into wild populations. And if enough of your population has these traits, then you will start to see population declines. And that is why stocking obviously eventually leads to forever stocking. So geography of these game farm genetic variants, wild and game farm mallards are mechanistically different. We are starting to understand that. Now we are trying to understand, when does a hybrid recapitulate wild? So we're looking at the same things, but amongst hybrid individuals. Individuals that we know are 50-50, 60-40, 70-30, 80-20, 90-10. When does that bird become mechanistically wild? So we can start to tell agencies what a cutoff is for what is wild. Wild mallard populations show very distinct patterns in genetics that is for now explaining behavior and population trends. Again, Great Lakes birds and Atlantic flyway birds have decreased by 50% over the last 10 years. And so, and in fact, the thing that we see in these populations is what is expected as you keep adding poorly adapted traits into a population. It's an additive effect. Increasing presence of game farm derived ancestry in Western locations since I've been studying. This rate is continuing to increase, to be fair. I've been doing this for 10 years, and everywhere I've been looking 10 years down the line has increased in frequency of these game farm genetics on the landscape. This will require further monitoring with updated maps that will be useful for management. We can get it to a particular wetland. How many hybrids? What is hybrid prevalence on one wetland versus another? If you can understand where the hybridization problem is, you could potentially have special seasons or tags or whatever it is that they decide to do. Where we're going with this to really get to the wildness is we've done full genomes of genetically vetted individuals. Again, that underlying theme, I know who I'm looking at. So I have ancestry of individuals, so we're looking at genes, and then each generation that gene moves or doesn't move tells me how strongly selected it is for or against from generation 1, 2, 3, and so forth. The other part of it is we are doing that whole history. We went to 29 museums. We have 700 historical samples across space and time from 1800 to today to understand how this happened and what this means for the conservation of mallards. And I never actually thought I'd ever have to say the conservation of mallards, a population that is, 14 million strong in North America, although I think we're at 11 million this year, BPOP surveys. But still, way better than some endangered species, right? I just want to acknowledge all my collaborators, Mike Schumer, Robert Krause, Paul Sundquist, for all the efforts to get all the samples across the board. But to be fair, there's no way for me to have done this work without basically every state, federal agency in the US, Canadian Wildlife Services in Canada, my Mexican partners in Mexico, partner New Zealand Fish and Game, Hawaii Fish and Game, and all the folks in Europe, all my students, and of course, funding. Can't do this without NSF, right? And all the other DNRs and state wildlife agencies. Before I thank all of you for listening to me ramble about mallards, if you are interested, our field site in American Samoa, we have a field site where we study Pacific black ducks. We do both habitat work, banding, we catch the birds, we band, put telemetry units, we do genetics, all that good stuff. If you ever wanted to catch a duck on a tiny, tiny island, come join my team, because we're looking for 4 to 5 techs. Pretty awesome, island life, it's really weird.